Good morning, बच्चों. Myself, Dr. Sunil Tavarkar, I am the biology faculty. So, biology is the subject, as you know, it is the related to the living organism. Okay, so study of the living organism is the branch of science that is called as the biology. Okay, and when we are talking about the living organism, so living organism having some properties, and one of the important property of the living organism that it is made up of the cells. Okay, so cellular organization, presence of cellular organization is the main character of the living organism. So as you know, the cell theory which was proposed by Sheldon and Shawn. Okay. So what the theory states that? The theory states that the cell is the structural and functional unit of the life. Okay, so each organism is made up of cell. The cell together form the tissue, tissue together form the organ, and organ form the organ system. And all the organ systems are together form the body of the organism. So cell is the structural unit of the life. Secondly, the function of the body is the working one. Okay, so the body shows the metabolism and all the metabolism that takes place within the cell. Okay, so the cell is functional unit of the life. So structural and functional unit of the life is nothing but the cell. Okay, the second point according to the cell theory is that each cell is developed or come into existing from the pre-existing one. जिस पहले से ही जो सेल प्रेजेंट है उनसे ही क्या होगा, नए सेल बनेगा. ओके, सो द न्यू सेल इज प्रोड्यूस्ड और गिवन बाय from the pre-existing cell. Okay, and that is by the process called as the cell division. That process is called as the cell division, means the pre-existing cell divide to produce the new cell. So that is the cell division. So when we are talking about the pre-existing cell, okay, means according to the virtue, it is omnis cellula e cellula. Omnis cellula e cellula means the cell is produced from the pre-existing one, or the cell is arise from the pre-existing cell. So that is called as the omnis cellula e cellula. So that process by which the new cells are formed from the pre-existing one, that is called as the cell division. Okay, so when we are talking about the cell division, so there are three types of the basically cell division. Okay, one is the amitosis, another is the mitosis, and the third one is the meiosis. Okay, so when we are talking about the amitosis, mitosis, and meiosis, so what happened there? In case of the amitosis, it is irregular division. Means from one cell, two cells are formed, but it is not sure that both of them are similar genetically, structurally. They are not similar. Then that is called as the amitosis. When we are saying mitosis, so what happens in case of the mitosis? From the diploid parent cell, two diploid are formed. Two diploid cells are formed. Or either the parent cell is haploid, it will produce the both haploids. Means from one cell, two cells are formed, which are genetically similar. These are genetically similar. So the equational division, mitosis is called as the equational division, as the chromosome number remains same. As the parent cell is diploid, the daughter cells will diploid. If the parent cell is haploid, the daughter cell will be definitely haploid. Okay, so the chromosome number will be stable, constant. That's why it is called as the equational unit. Okay, and the third one, when we are saying it is a meiosis, here what happens? The mitosis takes place in the somatic cell or the vegetative body cells. Okay, so here it leads to the growth. But in case of the meiosis, it is useful or it is significant in the production of either spores, that spores, microspores or the megaspores in case of the angiospermic plant, or it is useful in the formation of gamete. Okay, so when we are talking about the gamete or spores, it means meiosis takes place definitely into reproductive cell, that is called as the germ cells. Okay, and what happens here? After the meiosis, when the diploid cell, the diploid parent cell enters into the meiosis, from which what happens? 4 haploid cells are formed. 4 haploid cells are formed. So these haploid cells may enter the spores, microspores, megaspores, or in case of the animals, mostly it is useful in the production of gametes. So gametes are always haploid in women. Okay, but before going to the either mitosis or meiosis, the cell have to prepare itself for the division. Okay, and all that preparation takes place in under the successive stages. So all that stages that are included under the heading that is called as the cell cycle. Okay, so today we are going to discuss the cell cycle. So basically when we are talking about any division, so that division is having the two main phases, that is the young phase plus interphase. Young phase plus interphase. Okay, so when we are talking about the interphase, what is this? So earlier it is called as the resting phase, but today we can know it is not the resting phase, but it is having the higher activity, the stage of higher activity, that is the interphase, in which the cell will prepare itself for the next division. Okay, and this young phase, it either represents the mitosis or meiosis. Means the cell which is produced from the mitosis, that again enter into the mitosis, means to produce the new cell. Means it is the cell to cell. So it is called as the cell cycle. Means one cell that undergoes the mitosis, and by mitosis it again produces the cell. Means from cell to cell it is continuously going on. That is called as the cell cycle. Okay, so each cell cycle is divided into again. Okay, so this is the young phase plus G1, G2 and S phase. Okay, G1, G2 and S phase. Between this G1 and G2, actually this S phase takes place. Means if the sequence is corrected, it should be the G1, S and G2. And after G2, again the cell enters into the young phase. Okay, so likewise it is continuously going on. Okay, so here I will draw the cell cycle, and one by one we are discussing the stages in it. So this is the young phase, G1, S and G2. Okay, so these are the stages which are present in the cell cycle. Okay, actually to complete the cell cycle means mitosis plus interphase. So this interface takes about 95% time or the period that is required for the cell cycle, about from which 95% is required for the interface, where this interface includes G1, S and the G2 phase. Okay, and this M for the mitosis means actually cell division takes place. This is divided into the prophase, metaphase, anaphase and the telophase. Okay, so when the new cell is produced from the pre-existing one, that cell undergoes the resting period, but that resting having the multiple type of the activities or the metabolism, high rate of the metabolism can be seen in the interface state in the new cell. Okay, so this G1, firstly with the G1. So what is this G1? This is called as the gap phase 1. Or this is called as the growth phase 1. Okay. Now the name itself indicates growth. Means the new cell which is produced after the mitosis is obviously smaller in size. So it have to proliferate. Okay. So the proliferation, what happens? The proliferation of the cell takes place. Means the size of the cell increases in the G1 stage. Okay. Simultaneously in the metabolism, what happens? Transcription. Okay. By the transcription, what happens? The mRNA, okay, rRNA or the tRNA that are produced from the DNA. So that process is called as the transcription. Okay. So when the mRNA, rRNA or tRNA that are produced, once the RNA is there, so it will be useful for the production of protein, that is the protein synthesis is takes place. Okay, so in case of the G1, what happens? Firstly, transcription takes place. By the transcription, RNA are produced. By the RNA, protein synthesis occurs. Protein synthesis occurs. Okay, so why the protein is required? For the production of the new cell, of course. Okay, simultaneously the protein is also useful for the production of enzymes. Okay, so the enzymes that are produced with the help of protein which is produced in the G1 phase. Okay, so these enzymes are required for the S phase, that is the synthesis of the DNA. So all that enzymes are produced in the G1 phase. Okay, so these are the primary the metabolism that is takes place in the G1 phase. But sometimes what happens? From this G1, the cell comes outside of the cell cycle. Cell comes outside the cell cycle and it enters into the G0 stage. It enters into the G0 stage. What is the G0 stage? So that is called as the quiescent stage. Quiescent stage means there is no further cell division. So it directly goes to become permanent in nature. So when the cell is entered into the G0 stage or the quiescent stage, the cell will become permanent, it will never divide again. But if the cell from G1 enters into the S, means it is going to divide. Okay. Then the second stage, that is the S stage, S for synthesis. S for synthesis. So what is going to synthesis here? That is the DNA. The pre-existing DNA which are present in the divided cell or daughter cell, that undergoes the process of replication, and by the replication DNA is doubled. Means amount of the DNA is going to double. Okay, note down. If the parent cell is diploid, okay, means it having two set of chromosomes. Actually, it is nothing but the 2C volume of the chromosome. In the whatever the nuclear material present or the DNA material present, it is the double amount. Okay, but when the cell is going to divide means it have the again doubles its material, so it must be becomes 4C volume. Okay, means 4 times more volume will be there, so it will produce the 2 different cells. Okay, so the DNA volume it becomes 2C to 4C, 2X to 4X. Okay, so I am going to get doubles. Okay, but no chromosome number will be double. Chromosome number is same, only the genetic material or the DNA that gets double in case of the this S phase, that is called as the synthesis. Next to that, the G2 phase. Okay, so G for the gap phase 2. Okay, or we can say growth phase 2. Okay, in case of the G1 also the growth takes place. Whatever the remaining growth is occurs, that is into the G2 phase. Okay, so again the growth will be there, proliferation of the cell will be there, and cell organelles that also get doubles. Means if the cell is divided into the 2 and that 2 must be the equal or similar, then each and every material should be equally distributed into the 2 cells. Means not only DNA is distributed equally into the 2 cell of the cell, but also the different cell organelles into the mitochondria, into the chloroplast. As we know, these are the self-replicating bodies. Okay, so these organelles also doubles in case of the G2. Okay, then again protein synthesis takes place. Again, the protein synthesis takes place in the G2. So here the protein synthesis takes place, that is used as the enzymes for the process of DNA replication or duplication of the DNA. Okay, but here the proteins are formed, mostly the tubulin protein. Okay, the tubulin protein, it is the contractile protein and it is used for the formation of the spindle fibers at the time of cell division. Okay, so these spindle fibers can be contract, means these are the contractile protein. So such contractile proteins are also present, produced in the G2 phase. Now, okay, again, centrioles, doubles or duplicates. Okay, each cell having one centriole. Okay, but no doubt only animal cells having the centriole. The centriole are absent in the plant cell. Hence, the cell division in the animal cell is called as the centric cell division. Okay, but in case of the plant, as the centrioles are absent, it is the acentric division. Okay, so the centriole, it becomes 2 in number. As we know, the centriole that goes to the opposite pole in the cell, and from the centriole, spindle fibers are raised. Okay, which are to pull or push or pull the chromosome towards their respective pole. Thus, the chromosome number will be equally or half. Okay, so that distribution this depends on the spindle fiber. So for that, it must have the centrioles. Okay, so likewise, now the cell is fully prepared. Okay, firstly growth takes place, then RNA takes place. Okay, then protein synthesis occurs, that produce the enzymes used for the synthesis of the DNA or the replication of the DNA. Okay, so the DNA material of the genetic material, it will get doubles again, means 2C to 4C. Then in G2 phase, remaining growth takes place. The cell is now fully prepared by the formation of the cell organelles that get doubles. Okay, formation of the synthesis, tubulin protein are there, which are contractile protein used for the production of the spindle fiber, and the centrioles that also get doubles, means it becomes 2. Okay, So likewise now the cell after the G2 phase is completely prepared itself for the entering into the M phase. That M phase is nothing but the mitosis or meiosis. Okay. Now one more thing is there. As the G1 phase is comes after the mitosis. G1 phase is comes after the mitosis. So it is called as the post mitotic. It is called as the post mitotic phase. And as G2 it comes before the mitosis. So it is called as the pre-mitotic. It is called as the pre-mitotic phase. So likewise, this is the cell cycle, and here now it enters into the M, that is the mitosis, this having the prophase, metaphase, anaphase, telophase, in which at the successive stages the chromosome that gets divided into the two sets, and both these two sets that are converted to the two cells, the karyokinesis and cytokinesis is followed in this M1. Okay, and lastly again the two daughter cells are produced, which may either again enter into the cell cycle or that may withdraw themselves from the cell cycle and go to the G0 and becomes the organ. So this is the cell cycle. Thank you.